let's talk about yeasts. So these are the single-celled form that the fungi can take, and they can be different shapes. They don't all look like this, but what a lot of them can do is grow without oxygen. So you might be familiar with brewer's yeast and baker's yeast. Those can do fermentation. They do ethanol fermentation, and that doesn't require oxygen. One thing to remember is they replicate via mitosis, just like your cells do. And they can do this either in an equal way, where both daughter cells are the same size, sort of like what bacteria do, or they can do it in unequal form by budding, which is where the one daughter cell appears as a little tiny cell on the surface that elongates and grows and then splits off. And this looks simple, but it's more complicated than bacterial growth. And you might have guessed that because of what you know about cell division in humans. So sometimes what we will find in yeasts is that they don't detach from each other. And so when that happens, we'd call that a pseudo-hypha. We would say they formed pseudo-hyphy. But they're not true hyphy. And so let's understand what true hyphy are. So molds make true hyphy. And these are filaments. So if you look over here, what you see is a drawing of two different types of hyphy with a cell wall around the outside and nuclei. These little purple things are nuclei. And what these are for is invading new territory and exploring new territory. Because what they're designed to do is allow this cell up here, if it absorbs something good, it can share that with all the other cells. And so hyphae allow mold to maximize their surface area and exploit chemicals in the environment. They divide using mitosis the same way that we do. But what is crazy about hyphae, and what really makes them kind of elegant if you think about what's going on, is that each nucleus shares cytoplasm with all the other nuclei. So in this one, there's nothing separating this nucleus from this nucleus. They're all bathed in the same cytoplasm. And that allows molecules to diffuse, or more specifically, to be trafficked in vesicles long distances. In this one, it's more complicated because there are septa, which kind of break this into segments, but the septa have holes in them and are designed to allow cytoplasm to flow through. What we typically see, if you see a mold growing, what you're typically seeing is a mycelium, which is a large collection or network of hyphae. An individual hypha, generally speaking, is microscopic. It's too thin for you to really see it, but a mycelium could be an enormous collection of hyphae that altogether become visible. And what they're doing is sharing nutrients. So all these little white hyphae are sending nutrients back to the center. And what this is likely to do is this thing at the center is probably going to make some kind of a reproductive structure that will make little spores and grow up into the air and release them. And all these cells are working together to make that happen. So what conditions do we find fungi in? Now that you've seen their shapes, and of what determines where we find them, well, it's the same things for bacteria, but a little bit different. So water is always the most important thing, and water can allow some fungi to do things like eat wood. There are some plant compounds like lignocellulose that no microorganisms can digest other than certain fungi. So, and they can only do that when they can grow. And so water is something they're always looking for. If we go to the next slide, this is the same water activity table you saw from Unit 1. And I just want to point out that down here with some of the lower water activities, we see a lot of the fungi and not bacteria. These I believe that's archaea and that's bacterial, but the vast majority of bacteria are up here and a lot of fungi are down here. So they don't need as much water usually as bacteria do. That's a fair thing to say about the difference between them. In terms of food, I've alluded to this, but they can break down chemicals in like wood. And they do that to liberate simple sugars and amino acids. If they can break down lignocellulose, they get sugars from it. And they absorb those directly across their cell membranes. And that's what's meant by the absorptive nutrition of the fungi. Some of them require oxygen, sometimes they don't, and most of them are going to grow at typical outdoor temperatures. Fungi don't generally explore the extreme environments that bacteria and archaea explore. You don't find hyperthermophilic fungi or extremely acid tolerant fungi. Those are bacterial and archaeal specialties. Oh, right, and the last one is pH. A lot of fungi like slightly acidic pHs. So do with that information what you will, but I do hope you go back and look at it and think about where you have seen fungi in the world and does it line up with what I just told you. They have a complicated way of reproducing. So if you look closely here, what we see is hyphy, a network of hyphy, and two different cycles with this in the middle. There's the hyphy, they all get longer through mitosis, so we know that. But also they can do things like make a sporangium. They can work together to make this structure that lifts up some of the fungal cells into the air to let them spread. And what gets lifted into the air is daughter cells that are semi-dormant, called spores. And they'll fly away to another place when the wind pulls them, and wherever they land, they'll start making new hyphae. That's asexual reproduction. Sexual reproduction happens when the hypha, which has all the cells in the hypha are clones, if it bumps into another hypha that is the opposite sex, they will together make some kind of reproductive structure, and the spores they release are the result of sexual reproduction. So they make diploid zygotes, and then they go through meiosis and release the haploid spores, which can form hyphae. So that is that. I'll talk about that a little more. Mushrooms. I want you to know about mushrooms since you probably eat them. You should know what they are. Mushrooms are reproductive structures, and most of the fungi don't make mushrooms and can't. So mushrooms are very specific to a certain group of the fungi. The purpose of a mushroom is to grow these little things. So if you look on these gills here, if you magnify them with an electron microscope, so this would be scanning electron microscopy, you would see these rough surfaces on the edges, and this is beautiful scanning electron microscopy of one of these regions with false color. So they went in and colored these brown. Yeah, so again, this is some amazing scanning electron microscopy. You can see things like the little budding scars. And what we're seeing here are the results of that sexual reproduction. And so I'm going to walk you through the life cycle of these fungi because I want you to understand it. We can start with the mushroom. Up at the mushroom, it's made of cells with multiple nuclei, and some of the nuclei would be the positive and some would be the negative. Positive and negative are how scientists think of what I've been calling like female and male. There isn't really a female and a male. You can't see differences between the positive and the negative, so they just call them positive and negative, and here they draw them in different colors. So the mushroom is made up of cells that have nuclei with both of those types. And when opposite nuclei find each other, they'll fuse and make diploid cells, and those are the zygotes. And what that should tell you is that they were haploid. So everything here is haploid except this zygote stage. And they then go through something like meiosis. They go through the same divisions we would go through in meiosis, and they make these haploid spores. And those are these guys. Those are haploid little spores. And wherever the spores land, they will found a new mycelium or a new hypha. And that hypha will grow, and all those cells are clones of the original cell. So all of these cells are clones of this cell, and these cells are clones of this cell. Since they're haploid, they can only be one type, either positive or negative. And so as they're growing, if they come across the other type, if they grow across a hypha of the other type, they can join with it and have both types of nuclei in single cells. And once that happens, if they have enough nutrition, they will go through a complicated process of gaining enough nutrition that they can build a mushroom. And that mushroom will lift up these spores into the air where they can be released. And this allows them to do sexual reproduction, which is good for genetic diversity, and it allows them to lift up their spores a little bit higher so they can spread across the ground faster than they would just by mitosis. So that's what a mushroom is, and I'm happy to answer questions in office hours if that didn't make sense. Okay, so that's the basic, a lot of the basic fungus biology. Some cool things we see are what fungi do. Some of them are mutualists that live in plant roots. This is one of the cool things that they've been studying since I was in grad school, where fungi seem to act as supplemental roots for plants. They're better at getting nutrients out of soil. They do absorptive nutrition better than plant roots ever could. And so they can get nutrients out of the soil and give them to the plant roots. And the plants, which have endless food, since they just synthesize sugars from carbon dioxide, they can give sugars to the fungi. And so these associations between the fungi, the mycorrhizal, so rhizae would refer to roots and myco is fungal. Some of them live inside plant cells. And everywhere in nature, if you see lush growth of plants, I think we can guarantee you will find mycorrhizae in their roots. This is an experiment where they treated with different species of mycorrhizae. I believe the deal is that a plant will be adapted to a specific fungal strain, and so if you give it the right one, it will grow very well. If you give it the wrong one or none, it won't grow as well. And so this is something that people are studying in agriculture to try to help or find ways to grow crops faster and better. Another place we see fungi is lichens. And lichens are super cool. They are another symbiotic association where a fungus lives with a photobiont. A photobiont is not some new type of life you need to learn. It's just the random name they came up with for photosynthetic bacteria. So a lichen is where a fungus is going to grow with a photosynthetic bacteria. And so the fungus attaches to a surface, and its cell wall protects the bacteria. The bacteria provides sugar that it can make through photosynthesis. And probably the photobionts can live by themselves, but the lichens allow them to grow in different places. And you can read what nerdy evolutionary biologists say about maize or corn, where we live in a tight, humans live in a tight mutualistic symbiosis with corn. People my age, a big fraction of the carbon in their bodies came from corn. And so we say that we plant the corn and we bring it everywhere in the world, and it grows and gives us the carbon it fixes so nicely. I don't know. You don't need to think too much about corn if you don't want to. Just understand lichens. Lichens are important in some places. What they will do is break down rocks and start to add organic material to them, and that's the first step towards making soil in some environments. So if you have rocks, for example, that break off of a cliff and land on the ground, lichen might be the first thing to colonize them before they become soil. And animals will eat lichens. Some other cool stuff about fungi, as we will see, some of them cause diseases, either annoying ones like tinea pedis or athlete's foot, or deadly ones. But also fungi make drugs. So this is a painting that my boss used to have in her office that commemorates the discovery of the penicillium mold. So this mold that this person found in a, on some kind of melon. She was a lab technician who was looking for samples of mold, and she found one. That mold made penicillin. And so these researchers were looking for versions of the penicillium mold that could make that can make huge quantities of penicillin very efficiently. So this was back in the early 40s, and they were trying to make large quantities of penicillin. Because they were able to make large quantities of penicillin, World War II is the first war in which the majority of soldiers didn't die from bacterial infections. Because people with wounds could be given penicillin and it would protect them from a lot of the bacteria that otherwise would have caused deadly infections. So what I like about this painting, besides the fact that it was in my boss's office, was the fact that I grew up pretty close to this farmer's market, so I could go there if I wanted to. It's not still there, but it was when I was young. So isn't that fun? That's fun. The last couple things I'm going to say about fungi before we move on is remember that they can break down carbon sources that nothing else can. So this is an example of a place where there's been a flood and there's mold growing on the walls. Well, what is it using for carbon? What is the mold eating? It's not photosynthesizing, right? It's breaking down the wall materials. Bacteria weren't doing it. Animals weren't doing it. Only the fungus can break that stuff down and make it available to other organisms. And the same thing happens in forests with wood. If a tree falls down, we depend on the fungi to break down some of the wood. And then the last thing is, I have one more. Fungi make food. These are things like kombucha, mushrooms, yeasts that make the bubbles in bread, yeasts that make the bubbles in beer, some agricultural byproducts like marmite and nutritional yeast that are both from ability of byproducts from making beer. And then if you see a picture like this, what you should imagine is the roots of all these plants are happily growing with mycorrhizal fungi. Okay, so that's it for the beginning of lecture 5. What you're going to see after this are 3 more sections. One is going to be diseases and pathogens of the fungi. Then there's going to be a little bit of protist biology, and then diseases and pathogens of the protists. So that's where we're going with this lecture. And again, what's going to be on the upcoming quiz is the first 3 sections. So we just finished fungal biology. Now we're going to look at fungal diseases, and then we'll look at protist biology, and that's what's going to be on the upcoming quiz. Okay, I'll see you at the next video.